Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speaker in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speaker your questions. Your questions in the question and answer window will only be visible to myself and our speaker. So thank you for joining us today. It is now my pleasure to introduce our speaker, Dr. Amy Kauffman from Corning.
Good morning. My name is Amy Kauffman, and I'm a Senior Development Engineer at Corning Life Sciences. Thank you for joining me today as we're going to talk about how we can revolutionize acellular therapies with Corning Hyper technology. Here's a quick look at the agenda for today's webinar. We're going to talk about acellular therapies in the context of applying extracellular vesicles as therapeutic agents. First, we'll go through a brief introduction of what EVs are, how they can be applied for regenerative medicine, and what are some of the challenges. We'll then look at some of the data regarding the optimization of cell culture conditions to produce EVs in the Corning hyperstack. So let's dive in. Extracellular vesicles is an encompassing term for a population of particles naturally released from cells that are delimited by a lipid bilayer and they cannot replicate. EV are secreted by all cell types and found in nearly all biological fluids in the human body. The nomenclature surrounding EVs has been defined by the International Society for Extracellular Vesicles, which categorized subpopulations by biogenesis, or their release pathway, and their size. You're probably most familiar with the term exosome, which is the smallest vesicle with a diameter of an average of about 30 to 100 nanometers, and they originate from the endocytic pathway. At present, the most preclinical research focuses on EVs that are somewhere between 30 and 150 nanometers in size, regardless of their biogenesis. These are known as small EVs. This size range has demonstrated to be the critical carriers of paracrine factors such as proteins, cytokines, and RNAs. All functions of EVs have not yet been elucidated. However, it's very clear that they are mediators of intracellular communication. They're involved in many physiological responses, both in health and disease states. The small size and limited capture quantities of EVs from patient samples makes it challenging to understand all the critical roles that EVs play in biological regulatory activities. That being said, the composition of EVs is not random. The cargo delivers a distinct molecular message composed of proteins, lipids, nucleic acids, and sugars, in a unique pattern that's transmitted to the recipient cell to begin a response protocol. These response protocols can range from complex pathological conditions, such as metastatic cancer, to routine benign conditions, such as initiating wound healing of an epidermal cut. Now let's talk about how these vesicles could be used in research and in the clinic. When thinking about clinical applications of EVs, one of the first areas of interest is using EVs as diagnostic screenings via non-invasive procedures and testing protocols. As mentioned before, EVs are in every biofluid in the body, and technology has greatly advanced in the last decade to improve minimally invasive liquid biopsy techniques that allow clinicians to quantify disease biomarkers in EVs. The complement of biomolecules inside of an EV reflects the parent cell, and their characterization may provide information about the presence of an aberrant process. For example, peripheral blood is a rich source of circulating EVs, which is easily accessible through a patient's blood sample. An analysis of EVs in the peripheral blood could provide access to unparalleled amounts of biomarkers of great diagnostic and prognostic value. A second application of EVs in the clinic is using them as therapeutic agents. EVs possess inherent tissue repair promoting properties, and with their small biological design, may be an alternative to whole stem cell therapies. To give a little history, researchers initially thought that the cardioprotective properties of mesenchymal stem cells, or MSCs, resulted from their differentiation into healthy myocardium. Through rigorous experimentation, it was determined that the effects were actually paracrine in nature and not the direct application of MSCs on the surrounding tissue. Significant interest in the therapeutic potential of EVs was then generated as it was demonstrated as they are the important component of the MSC-mediated cardioprotection pathway. Since this discovery, extensive preclinical research has revealed the utility of EVs as a therapeutic agent in other organ systems as well. One of the many reasons EVs are successful therapeutic agents is that they possess numerous advantageous features as drug delivery vehicles that may help them outperform synthetic drug carriers. Notably, EVs seem to possess an intrinsic ability to cross tissue and cellular barriers. In addition, synthetic drug carriers such as lipid nanoparticles or polymeric micelles suffer from high immunogenicity and toxicity. As therapeutic EVs are derived from either autologous or benign biological sources, they are less likely to induce these adverse effects. Furthermore, some EVs may possess inherent targeting characteristics and display tropism for a particular cell or tissue type. This feature could be exploited to selectively deliver drugs to their intended targets while avoiding off-target effects. Let's take a look at the difference between EV and whole stem cell therapies to understand some of the opportunities EVs present to the challenges of whole cell therapy. The workflow for whole stem cell therapy in the clinic can be challenging and time-consuming for both the patient and the clinician. The first step is acquiring blood from the patient or a donor. Viable stem cells are then separated from other cell types and blood components via centrifugation. The separated stem cells then go through activation, which provides the appropriate stimulus to program cells to ensure they will migrate to sites of inflammation and exert anti-inflammatory responses. The activated cells are then given or returned to the patient as the final step of treatment. As you would expect, there are both technical challenges and safety concerns that explains why there is limited stem cell products that are FDA approved and only a few that are operationally approved. The biggest technical hurdle is the quality of the patient or the donor's cells. Often patients have already progressed significantly into the disease where cell health, heterogeneity, and genetic instability can be in question. If the patient is also the donor, multiple procedures are required for the individual. Lastly, delivering whole live cells to a patient comes with risks both in their size and their functionality. Unfortunately, a patient's immune system can reject donor cells, or cells can coalesce and propagate into tumors. Cells may also not have achieved optimal activation, which could result in cell migration inhibition and functional failure. In contrast, EVs possess numerous advantages over cell-based therapies in the context of regenerative medicine. A major advantage is that EVs, depending upon their source, may be less immunogenic than their parent cells. Immortalized cell lines, including mesenchymal stem cells, can serve as donor cells for in vitro EV production. In this case, activation is also likely not required, as the source of therapy is the paracrine factors that are delivered by the EVs. Unlike live cells, EVs can have a long shelf life and may be transported and stored for long periods of time. The nano size and natural biologic design of EVs allows for their stability and stealth protection from the immune system to prevent treatment rejection. Furthermore, EVs do not replicate after injection. Thus, EVs present less risk of tumor generation and transfer of latent viral pathogens. It is not expected that EVs will need to be produced from the patient's donor cells. This would allow for the patient to only need to be in the clinic for the receipt of treatment, and they would not have to have multiple handling steps of the patient's cells and subsequent EVs in the hospital laboratory. Despite the potential advantages that EVs have over whole stem cell therapies, challenges remain that prevent EV therapeutics from translating from bench to bedside. First is the lack of bioprocessing methods for scale-up of MSC-derived EVs. The second is being able to translate the appropriate conditions from human physiology to in vitro cell culture to support the intrinsic ability of MSCs to produce high quality and quantity EVs. Currently, there is no consensus on the optimal technology for EV production. Common protocols for the production of EVs are mainly based on 2D static cell culture platforms. It is well understood that there are fundamental differences between 2D adherent cell culture and normal cell physiology in the human body. However, we can exert control over the environment in a cell culture platform that can help guide the physiological processes necessary for quality EV production. As explained extensively in the literature, many environmental factors, both chemical and physical, in the body can stimulate EV production, and we can mimic these in vitro. Mimicking chemical changes such as oxygen concentration and food deprivation can have detrimental effects on cell health and viability, and the subsequent quantity and quality of EVs may suffer. For our studies, we chose to mechanically stimulate 2D adherent cells with fluid movement in a dynamic culture system, which is expected to have minimal impact on cell health while increasing EV production. The movement of fluid is being used like a clearance mimic, where the cells may be induced to produce more EVs when those pericellular EVs have been relocated away from the sensing regions of the cell. Our work completed 2 independent studies where Vero cells were scaled and cultured in serum-free conditions using the Corning HYPERStack 12 vessel. The vessels were divided into 2 mechanical condition groups, static being our control, and dynamic, which we have the HYPERStack 12 placed on an orbital shaker operating at 30 RPMs directly after seeding. After seeding the HYPERStacks, the Veros were cultured for 72 hours. The spent media was collected, and EVs were isolated and concentrated using ultracentrifugation. The collected EVs were then characterized using nanoparticle tracking analysis and a colorimetric protein assay. Remembering that our first study was seeded with 11,000 cells per centimeter squared, and our second study with 17,000 cells per centimeter squared, we can see a healthy expansion of Vero cells over 72 hours. Cell viability averaged above 97% for each vessel, regardless of static or dynamic culture conditions. Now looking at EV production, we observed that the dynamic media conditions increased EV production on an average of 56% greater than the respective static vessel control. Looking at the right graph, you might have expected to see the bars of the second study to be greater than those of the first study. In this calculation, the number of EVs were normalized per million cells. This way, we can look directly at the influence of dynamic media culture and eliminate the effect of different start seeding densities. Let's briefly take a look inside of the Corning HYPERStack and why it supports the expansion of adherent cells. Each layer of the HYPERStack has an ultra-thin gas-permeable film that is spaced between rigid polystyrene layers for support. The enhanced gas permeability allows for scaling of cells in a small footprint. Recently, a Corning application note published successful expansion of MSCs with high viability and no impact on normal stem cell characteristics. Successful scale-up of MSCs and enhanced EV production with optimized cell culture conditions show promise for large-scale MSC-derived EV production. In conclusion of this webinar, I hope you can take away a better understanding of extracellular vesicles and their application as acellular therapeutics in regenerative medicine. EVs are naturally produced intracellular communication biologics that offer an alternative approach to whole stem cell therapies. There are still technical hurdles, such as establishing bioprocess methods for scale-up and cell culture condition optimization that needs to be addressed to bring these therapies viably to the clinic. Today, I shared results of our study of the effect of altering cell culture conditions from static to dynamic in the Corning HYPERStack cell culture vessels and how the subsequent production of EVs can be increased greater than 50%. This proof of concept work is a stepping stone in exploring Corning bioproduction vessels for enhanced large-scale EV production and a future of possibilities for acellular therapies. I would like to take this opportunity to acknowledge my colleague, Pilar Pardo, for her help with the experiments I presented to you today. I would also like to express my gratitude to BioProcess International for the opportunity and thank all the listeners for tuning into today's webinar. At this time, I'm happy to answer any questions you may have, or please feel free to reach out to me via the email address on this slide after the webinar. Thank you.
Okay, Amy, are you ready for your first question?
Sure am.
Okay, so the first question is, for EV therapies, it appears that EVs derived from MSCs would be the most valuable. Why were Vero cells used in these experiments?
Right. As we mentioned, all cell types produce EVs, and there's going to be varied benefits from different cell types and the EVs that are produced from them. In this case, we chose Veros, one, because they grow really well in serum-free conditions, so it's easy to separate any sort of noise that can happen from using serum in conditions, since that can contain a variety of EVs already in there. Also, they are an interesting cell population that is being used for viral transduction studies, so this could also be interesting for the current global pandemic setting. But yeah, they're a really great model to work with, and it's a good place for us to start.
Have you investigated other EV production enhancement techniques beyond mechanical?
We have, and one of the things I did mention is a lot of the chemical products that you can use in order to enhance production can also have an effect on the overall health of the cells. So we've looked at a couple different additives. We've also looked at some of the starvation techniques, which again, we do see a decline in cell health over time, and we're a little bit concerned with the EVs that come out of cells that are not at their highest health.
And were the seed train cells in the hyperflask also cultured under dynamic and static conditions? And do the cells need to be accustomed to the dynamic conditions?
Right. So in this case, in the seed train, they were not under dynamic conditions when they were grown up from T-flask to hyperflask to the hyperstack. They were only under dynamic conditions once they entered the hyperstack as a whole. For our study, we did not have a customization step where the cells were allowed to settle and then the dynamic conditions were used. We actually went straight into dynamic right after seeding. We found with a couple of experiments that the cells don't actually need an acclimation period and are very active as soon as they hit the dynamic condition.
Okay. Well, it looks like we have time for just one more question. If you didn't get to your question, we will be passing this on to Amy directly. So please continue to type any questions we have, and we will pass those on. But the last question for the webcast is, are there any plans to investigate Corning perfusion vessels for EV production?
Yes, so this work started small where we began in T-flasks and have grown up now to something as large as the hyperstack, but there is ongoing work in the cell cube, as well as there are plans for looking at EV production out of Corning's bioreactor, the Ascent. We're also looking into other 3D culture platforms, so everything we've talked about today, we're 2D culture, but there'll be more information for that coming soon.
Okay. Thank you, Amy. And thank you to our audience for joining us. The recorded version of this webcast will be available for on-demand viewing on our website, and as a registered attendee, you'll receive a follow-up email providing you with a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.